Most of the early studies on fermentation were done on various single-celled Ascomycetos fungi of the genus Saccharomyces, what we commonly refer to as yeast. Yeast became an international house staple with its presence in wine, beer, and bread. It can trace its roots to the Sanskrit word yazyati, meaning boils or seethes, since the bubbling and foaming of fermenting beverages closely resembles that of boiling. Yeasts are chemoorganotrophs. They use organic compounds such as hexose sugars and disaccharides as a source of energy and do not require sunlight to grow. Carbon dioxide, water, and ethanol are but a byproduct of their digestion. Yeasts are considered to be facultative anaerobes, having both aerobic and anaerobic methods of energy production. The specific species of yeast we will be discussing, the sugar-eating fungus Saccharomyces cerevisiae, is known as baker's or brewer's yeast. It is round, about 5 to 10 micrometers in size, and multiplies through an asexual reproduction process known as budding. They are so tiny that about 1 gram is around 25 billion cells. So how did we discover something so small? Some say it was most likely 5,000 years ago, with dough that had been sitting around for far too long, accumulating natural yeast organisms. Others look to when wine was first encountered, about 7,000 years ago, in the Neolithic period. It was as simple as a fruit jar, sitting, fermenting, and the rest became history. What we do know is that for centuries, cultures have been using beer and wine as a source of yeast to leaven and lighten bread. Yet however it was discovered and used, it was not observed until the 17th century with the emergence of the microscope, and not considered to be living until 150 years after that. Finally, in 1857, Louis Pasteur demonstrated that yeast cells can live with or without oxygen, and that these microorganisms were directly responsible for alcoholic fermentation and dough leavening. Now let's return back to the metabolic process of yeast to better understand what these microorganisms are doing. As we mentioned, yeast, the fungus with a sweet tooth, feed on sugars and starches, turning them into energy. In bread making, the yeast initially respires aerobically, releasing carbon dioxide gas and water as a metabolic byproduct. It is the pockets of gas produced by yeast that causes the dough to rise before baking, and it is what makes a slice of bread so soft and spongy. When oxygen is exhausted, yeast will respire anaerobically, and fermentation will begin, producing ethanol as an additional waste product. Ethanol will evaporate during the baking of bread, but is the foundation of alcoholic beverages. Mead, wine, beer, distilled spirits, even the lightly fermented kombucha, all use yeast at some point in their production. Yet in high concentrations, ethanol is toxic to yeast, as is the heat microbes produce during the metabolic process. So it's important to keep in mind that there is a limit to alcohol content in natural fermentation. All microorganisms go through a bacterial growth curve, and once yeast has reached a critical alcohol concentration, the yeast cells will start to die off. Though some superstrains have been developed to increase alcohol content, most die at 10 to 20% concentration. Now that we've explored yeast fermentation in bread and alcohol, let's observe some other forms of fermentation in food production.